So the plant growth regulators are chemical substances that influence the growth and the differentiation of plant cells, tissues, and organs. So this process we are talking about, where we have our zygote highly having cell division, causing growth in size, in width, in mass, in volume, and then differentiation of plant cells into specialized cells such as the leaves, the roots, the stems, into tissues and the organization of plant organs is usually facilitated by chemical substances, which we call hormones, phytohormones, or plant growth regulators. So these plant growth regulators function as a chemical messenger for intercellular communication, communication between cells, and they work together coordinating the growth and development. So they could be indoor compounds such as indole-3-acetic acid, which we call IAA. It can also be adenine derivatives. It can be derivatives of carotenoids like abscisic acid, which we call ABA. It can be terpenes like gibberellic acid, and it can be gases such as ethylene. So this is chemistry. Go and look out the various classification of these phytochemicals. This is supposed to be in the unit of biochemistry. So the phytohormones can be classified into these 5 major categories. We have auxins, we have gibberellins, we have cytokinins, we have abscisic acid, and we have ethylene, which is a gas. Now, these phytohormones, again, can actually be classified into either growth promoters or growth inhibitors. And for the growth promoters, we have auxins, gibberellins, and cytokinins. And then for those that inhibit growth, we have abscisic acid and ethylene. So we look at each of them. We start with auxins. Auxins were discovered by Charles Darwin and someone called Francis. So these guys isolated auxins from the tips of oat seedlings, Of course, the tips have high cell division, and that is why they were able to isolate auxins. And they went and developed a vena curvature test for bioassay. So there is this test for bioassay, which you can use to do a bioassay of the amount and presence of auxins. So we have synthetic auxins. Synthetic now means today, we don't just rely on auxins that are natural. We also have those that you can buy, because as biotechnologists, we need to promote growth at one point or another. Maybe you're growing a certain plant or you're doing breeding. So in this case, we are saying that if you want to promote growth, we have some forms of synthetic auxins. We have IAA, we have IBA, we have NAA, and we have 2,4-D, okay? These are the 4 examples of synthetic auxins. So auxins generally have the following effects. One of them, promote apical dominance. And apical dominance means that there is a shoot that is always growing, that is a favored growth, as others are forming stems. So if this is our major stem, others are forming branches. That's our plant. These are the roots. There is promotion of apical dominance. So this apex continues to grow, and that is what now gives length, continuous growth of length of plants. That is promotion of apical dominance. Initiating rooting in stem cutting. So if you cut a stem, of course not of every plant, but there are plants and flowers that we commonly just cut the stem, go plant, and it will develop roots. So those plants and flowers that we do that, what initiates this kind of growth and rooting, to have roots so that now it can grow into a plant, is what we call auxins. Of course, it promotes flowering in plants like pineapples, and then it prevents fruit and leaf drop at early stages. So this means it prevents aging, okay? So we prevent aging of plants before they have reached maturity. And of course, they promote abscission of older, mature leaves and fruits. Abscission means falling off. So if you have very old, mature leaves and fruits, they will fall off so that new ones can be propagated by the auxin hormone. Then they induce what we call parthenocapy, the development of fruit without fertilization, meaning we get seedless. And of course, they control xylem differentiation and helps in cell division. Of course, meaning it helps in the maturity of xylem vessels that are important for the transportation of water. Then we have gibberellins, and these ones were discovered by a scientist called Kurosawa, and he identified them present in a fungal pathogen called Gibberella fujikuroi, and that is why he called them gibberellins. So they were isolated from infected rice seedlings when treated with sterile filtrates of fungus. So usually this hormone facilitates elongation of intact stems. So we have had growth that has already been promoted by auxins. So the stems now will continue to elongate because of the presence of this hormone. Then of course it will dwarf shoots. Dwarfing shoots means that it doesn't promote elongation like we said of apical dominance. Remember we said auxins promote apical dominance, such that the tree keeps growing tall and tall. So for this one, it usually promotes more growth of the sides and not of the sides upwards. It is more of branches and shoots remaining down here. And what this does, it makes what we call, if we were to use it in layman, it will create a bushy plant. So that if it's a tree, instead of being thin with very little branches that way, it will be a tree that looks like this, okay? So this usually causes dwarfing of shoots so that they concentrate within a shorter area. And these days we have such kind of interventions in biotechnology, like when you talk about coffee, the new brands of coffee, those coffee plants are actually not growing very tall. You can actually just, pick coffee very easily because of the dwarfing of shoots by such PGR that is being applied artificially. We have bolting, the production of flowering stem on crops before the crop is harvested, for example, in cabbage. So this is not a good thing, particularly if it happens in cabbage, for example, because it means that it causes flowering in cabbages before they are harvested, and this will, of course, reduce the value of our cabbage. So if it is in excess, then gibberellins are not very advisable at that point. Then they overcome natural dormancy of buds, tubers, and seeds. So if there is a natural dormancy, maybe we have buds, tubers, and seeds that are not growing because there is no correct environmental conditions, gibberellins can move them to bud, can move the seeds to germinate. Yes, so seed germination. Then of course they induce parthenocapy or development of seedless fruits, just like auxins. They also promote flowering in long day plants, and they also promote vernalization, that is flowering due to exposure to very low temperatures. Cytokinins, they were discovered by Skoog and Miller, and they were able to crystallize the cytokinesis promoting active substance and named it kinetin from corn kernels and coconut milk. So this is where they were able to isolate and crystallize this product. The physiological effects of cytokinins is that they induce cell division, and that's why I said it's also a growth promoter. And of course, they delay senescence of leaves and organs, meaning they delay aging. They promote lateral budding, lateral on the sides, so similar to gibberellins, lateral. The other one was apical. Auxins were apical. So these ones are more lateral. And then increases cell expansion in dicotyledons and in leaves. Cell expansion. Remember we said cell expansion was part of a characteristic of growth. And then they promote chloroplast development and chlorophyll synthesis. Very important for the process of photosynthesis. And then we have now the ethylene. Ethylene usually is volatile, it's gassy. So it was, the Cousins confirmed the release of volatile substances from ripened oranges and hastened the ripening of stored and ripened bananas. So this volatile substance is produced by ripening fruits. And that is now the one that is being used today in biotechnology too. You know, you expose your bananas, you expose your avocados, you expose your oranges to this gas that is artificially made nowadays so that you can facilitate fruit ripening. Of course, it has been overused in some cases because you also need to allow the natural process of ethylene gas to be able to ripen our fruit. So we need to have them at least first, they have matured, and then the process of ripening. So this is a process that has been highly, highly contested today, and it is affecting the idea of biotechnology in most people because they know that this gas today is being misused, and you get avocados that are ripened on one end and they are not ripened on the other end because they have been manipulated to hasten. So ethylene gas is important, but it should be used when the fruits have matured to even make it easier for uniform ripening. So of course they promote senescence. So these are growth inhibitors. So senescence means the aging, and of course fruit ripening is part of aging. And then they also promote shedding of certain parts of plants. They inhibit the elongation. Now we are no longer talking about increase in size and volume. We are saying now the growth in length has come to an end. They, of course, they induce fruit ripening. They will break seed and bud dormancy. They will initiate germination of peanut seeds. They will cause the sprouting of potato tubers. Then they will also promote root growth and root hair formation. So this is now when our plants have matured. So it is more of differentiation more than increase in size. And of course, they also initiate flowering and for synchronizing fruit set in pineapples. That is the ethylene gas. And the other one is what we call abscisic acid that was discovered quite early by Frederick and his co-workers. And it was isolated from several abscission accelerating substances from cotton plants. And it is usually referred to as a stress hormone. So usually it acts as a general plant growth inhibitor, okay? And it will induce seed dormancy. It will inhibit the germination of seeds. It will stimulate closure of stomata. And once they close, it means photosynthesis is reduced or will no longer be there. And of course, it plays an important role in seed development, maturity, and dormancy.